What if the key to healing the intense physical trauma of childbirth isn't found in a surgeon's scalpel or even in live stem cells, but in these invisible microscopic bubbles? Bubbles
Mhm.
that basically act like tiny flash drives to rewrite the code of your damaged tissue.
It sounds like science fiction, honestly, but it's very real.
Right. And for you, the listener, especially if you're trying to catch up on the absolute cutting edge of regenerative medicine, today's deep dive is going to completely flip how you think about biology and healing.
It really changes the entire paradigm.
It does. We're looking at a fascinating breakthrough in how the human body can heal from severe mechanical trauma. Specifically, we are diving into how we can reverse mild to moderate introital laxity after childbirth and doing it without relying on live cell therapies. So, okay, let's unpack this.
Well, to really appreciate the scale of this breakthrough, we first need to look closely at the sheer physical toll childbirth actually takes on human tissue. I mean, we tend to think of the body as incredibly resilient, which, it is, but
But the strain is just off the charts, right?
oh, extreme. The mechanical strain placed on the pelvic region during birth is just immense. A baby passing through the birth canal can stretch the surrounding muscles and tissues up to, like, 3 times their normal length.
Wow. 3 times.
Yeah, and this causes profound damage at a microscopic level, specifically to something called the extracellular matrix, or the ECM.
Okay, the extracellular matrix. I always hear that term in like anti-aging skincare commercials, but what does it actually look like inside the pelvic floor?
Think of the extracellular matrix as a dense, three-dimensional netting. It's made of proteins, mostly collagen and elastin, so it's literally the structural scaffolding that holds the vaginal walls and the pelvic floor muscles in place.
Right, the support system.
Exactly. And during birth trauma, that netting is severely degraded. You see this massive drop in the production of new collagen, and the existing fibers are just overstretched, or they outright snap.
Ouch. And I imagine it's not just the scaffolding, right? Like the actual muscles take a hit too.
They absolutely do. The major pelvic floor muscles, like the pubococcygeus and the levator ani, they often suffer micro tears. And in more severe cases, they sustain full avulsion injuries.
Wait, an avulsion injury, that means the muscle is actually torn completely away from the bone, right?
Yeah, that is the clinical definition. It's a severe structural failure. And because the anatomy there is so tightly packed, the nerves in that area, particularly the pudendal nerve, they're stretched far beyond their physical limits.
Which causes its own set of problems.
Right, because that stretching can literally strip away their protective coating or even sever the internal nerve fibers completely.
And so all of this structural damage combined, that is what leads to that diagnosis of introital laxity, like a physical looseness or loss of tone in the vaginal opening and canal, which, often brings along stress urinary incontinence too.
Exactly. The tissue just can't support the normal function anymore.
Right. Now, looking at the history of regenerative medicine, for the longest time, the proposed solution for this kind of severe tissue damage was always live stem cells.
Yes, that was the golden ticket for years.
The idea being you just inject these master builder cells directly into the damaged area and they just, figure out how to fix everything. But that narrative seems to have hit a wall in reality. So if stem cells are the ultimate builders, why not just use them directly to fix the laxity?
Well, I mean, it comes down to a major biological bottleneck. We found out the hard way that deploying live, multiplying cells into a patient's body is highly, highly unpredictable.
Unpredictable how? Like they don't do what they're told.
Worse, when you inject foreign stem cells, you run headfirst into the problem of immunogenicity. The patient's immune system basically acts like a hyper-aggressive biological security checkpoint.
Oh, so it sees them as a threat.
Exactly. It scans the surface proteins of those live stem cells, recognizes them as foreign invaders, and just attacks them before they can even begin to heal the tissue.
So the body's own defense mechanism essentially destroys the treatment right out of gate.
Frequently, yes. And there is a second, even more serious risk, which is tumorigenicity.
Meaning tumors?
Right. Live stem cells are literally defined by their ability to rapidly divide and grow into different types of tissue. So if you place them into a highly inflamed, traumatized environment, like a postpartum pelvic floor,
The signals are all messed up.
exactly, the chemical signals are totally chaotic. Without perfectly clear instructions, those stem cells can just start dividing uncontrollably and literally form tumors.
Wow. That sounds incredibly dangerous. Plus, I imagine, just keeping a vial of live cells alive from a laboratory to a clinic is its own logistical nightmare.
Oh, it's a massive headache. It requires an unbroken cold chain of cryogenic freezing, specialized incubators, highly trained lab techs on site. I mean, the cells are incredibly fragile.
So they just die easily.
Yeah, a significant percentage of them just die in transit. Or they die shortly after injection because that inflamed pelvic tissue is simply too hostile an environment for them to survive in.
Okay, wow. This raises an important question. How do we get the regenerative benefits of stem cells without the severe risks and logistical nightmares of using live cells?
And that pivot brings us directly to the star of today's deep dive, the exosome. Scientists realize that, hey, if the cell itself is too risky and fragile, maybe we don't need the cell at all.
We just need the messages it sends out.
That is the fundamental leap in logic right there. Researchers began studying how stem cells actually communicate with damaged tissue, and they isolated these incredibly tiny extracellular vesicles. We call them exosomes.
Tiny is probably an understatement, right?
Oh, absolutely. To give you a sense of scale, an exosome is only about 30 to 150 nanometers in diameter. For context, a single strand of human hair is roughly 80,000 nanometers wide.
Okay, so they're unimaginably small, but where do they actually come from if they aren't cells themselves? Like, are they just floating around?
They are actually secreted by stem cells. So whether we were talking about adipose-derived stem cells, which are taken from fat tissue, or even urine-derived stem cells, the parent cell pinches off a tiny piece of its own outer membrane to create a little bubble.
Like blowing a bubble from a wand.
Pretty much. And it fills that membrane-bound nanobubble with a dense payload of bioactive molecules, things like messenger RNA, microRNA, proteins, and growth factors, and then it just releases it into the surrounding fluid.
Okay, so I'm picturing this. If a stem cell is a big, bulky desktop computer, an exosome is like a tiny USB flash drive. You don't need to transport the whole giant computer into the damaged vaginal tissue. You just plug in the USB drive containing the repair software and let the local cells read it.
That is a highly accurate way to visualize it. When you inject these exosomes into the damaged pelvic floor, they just drift through the tissue until they bump into the damaged local cells.
And then they plug in.
Yeah, because the exosome is made of the exact same lipid membrane as the local cell, they easily just fuse together. The exosome acts just like that flash drive, literally spilling its genetic code and protein software directly into the damaged cell. It completely reprograms its behavior.
And because it's just a bubble full of code, it totally bypasses all those live cell bottlenecks we just talked about.
Completely bypasses them. Exosomes have no nucleus. They do not have the cellular machinery to divide or replicate, which means they physically cannot form tumors.
Oh, that's huge. And what about the immune system?
They lack the major surface proteins that trigger immune rejection, so the body's security checkpoint just ignores them. They slip right past.
And I'm guessing they're easier to store, too.
Much easier. Because they are essentially just lipid bubbles filled with code, they can be freeze-dried in a process called lyophilization. So they can literally sit in a vial on a clinic shelf at room temperature for months without dying.
Okay, I'm really starting to see why this is such a massive shift in the field. We have these stable, microscopic USB drives, but what I want to understand is the software itself. Like, when these exosomes are injected into a patient dealing with introital laxity, how does a chemical message actually rebuild the scaffolding of the vaginal wall?
Well, the primary target for these exosomes in the pelvic floor is a specific cell called the fibroblast. Fibroblasts are the cellular factories that are responsible for spinning the collagen and elastin fibers that make up that extracellular matrix.
The netting we talked about earlier.
Right. But after birth trauma, these factories are essentially shut down, or they're just totally overwhelmed. So when the exosome fuses with the fibroblast, it delivers very specific microRNAs.
Now, I want to pause on that word. What exactly is a microRNA doing in this specific context?
You can think of a microRNA as a genetic stop-and-go sign. The exosome carries very specific signs. For instance, the research highlights a microRNA called MIR320A3PP.
Catchy name.
Right. But when this specific code enters a fibroblast, it acts as a massive green light for a gene called COL1A1. Now, this gene holds the exact blueprint for type I collagen.
So it basically yells at the factory to start working again.
It forces the factory into overdrive. It starts pumping out fresh, highly organized type I and type 3 collagen, along with elastin. And that is what restores the physical stretch and recoil to the lax tissue.
Okay, so that handles the building side of things, but I assume there's a reason the body wasn't just building this collagen on its own in the first place.
There is a very good reason. After a mechanical injury, the body releases these enzymes called matrix metalloproteinases, or MMPs, specifically MMP1 and MMP2.
Okay, what do they do?
Their normal job is to clear away damaged tissue by breaking down collagen. But in traumatized tissue, these enzymes go into overdrive. They keep degrading the collagen faster than the body can rebuild it.
Oh, so it's like a demolition crew that doesn't know when to quit.
Yeah.
The scaffolding is actively being dismantled while the body is trying to heal.
Yes, precisely. And this is where the second half of the exosome software comes in. They deliver another specific microRNA called miR-93.
And let me guess, that's the stop sign.
You got it. This molecule acts as a highly targeted stop sign. It chemically binds to the cellular machinery that produces these MMP enzymes and just shuts it down. So the exosome is simultaneously halting the destruction of the old collagen while forcing the rapid production of new collagen.
That is amazing. Okay, so the exosomes fix the scaffolding by swapping out the stop-and-go signs. But wait, collagen is just the scaffolding, right? If the actual pubococcygeus muscle was torn or heavily stretched during delivery, collagen won't help the muscle contract and function. How does a chemical message stitch a broken muscle back together?
It does that by targeting a completely different set of cells. Scattered along your muscle fibers are these dormant cells called satellite cells. You can think of them as a reserve military unit that stays asleep during peacetime.
Okay, so they wait for an emergency.
Right. In a normal, minor muscle tear, a few of them might wake up to help repair the damage. But birth trauma is often so severe that the local signaling pathways completely fail to activate enough of them.
So the reserve unit just sleeps right through the massive emergency.
Exactly. But when exosomes are introduced, they contain specific proteins that bind to the receptors on these sleeping satellite cells. And this triggers an internal chemical cascade, relying on specific lock-and-key pathways inside the cell, like the ERK and NFB pathways.
And what do those pathways actually do?
The exosome chemically unlocks the cell's ability to divide. So the satellite cells wake up, rapidly multiply, and then physically fuse with the torn muscle fibers to bridge the gaps and thicken the muscle tissue.
Wow.
Yeah, this is what drastically improves the physical tone and the resting pressure of the pelvic floor. It actively reduces the laxity.
If we connect this to the bigger picture, collagen and muscle fibers, even if they are perfectly rebuilt, they still need plumbing and wiring. Like a newly patched muscle is completely useless if it doesn't have oxygen, and it can't function if the nerve controlling it is severed.
That is a crucial point to remember. Building tissue without a supply chain is a huge waste of biological energy. So let's look at the plumbing first.
Okay, the blood vessels.
Right. Exosomes carry a specialized software program in the form of a protein called vascular endothelial growth factor, or VEGF. When exosomes release VEGF into the damaged vaginal tissues, it binds to the cells lining the existing damaged blood vessels.
And what does that signal tell them to do? Build more pipes.
Exactly that. It commands them to begin a process called angiogenesis. That is the sprouting of brand new blood vessel capillaries. The cells literally branch out, almost like the roots of a tree, growing directly toward the areas of the tissue that are starved for oxygen.
So it's physically building a new supply line.
Yes, and this new vascular network floods the newly synthesized collagen and regenerating muscle fibers with the actual nutrients they need to survive and mature.
Okay, that covers the plumbing beautifully, but what about the wiring? Because nerves are notoriously difficult and frankly incredibly slow to heal. If a woman's pudendal nerve is severely stretched during delivery, leading to, a loss of sensation or bladder control, how does an exosome speed up that specific repair?
For nerve repair, the sources highlight exosomes derived specifically from Schwann cells. Now, Schwann cells are the natural support system for our nervous system. They literally wrap around nerve fibers like rubber insulation around a copper wire.
Okay, I can picture that.
So when an electrical nerve fiber is crushed or severely stretched, the nerve cell often receives a chemical signal telling it the damage is just too severe, and it initiates apoptosis.
Apoptosis is programmed cell death, right? The cell basically just self-destructs.
Yes, the body basically prefers to cut its losses and kill the damaged nerve cell rather than expend the energy trying to fix it. But when a Schwann cell exosome fuses with that damaged nerve, specifically targeting these cells called DRG cells, it drops off a genetic code that physically blocks the self-destruct signal.
Wait, really? It just overrides the system?
Completely overrides it. It works through something called the wunderkatenin pathway. It stops the apoptosis command and forces the nerve cell to stay alive. And once the cell is stabilized, the exosome provides growth factors that encourage the nerve fiber to slowly extend and reestablish its electrical connections.
I am trying to visualize all this happening at once in a traumatized pelvic floor. We have, New collagen forming, muscles stitching themselves together, blood vessels sprouting, nerves regenerating. But there is one major hurdle we haven't touched on yet.
The environment itself.
Exactly. A pelvic floor after a difficult delivery is highly, highly inflamed. And everything I've ever read says that chronic inflammation stops healing in its tracks.
It absolutely does. And the mechanism exosomes use to solve this might actually be the most elegant piece of the entire puzzle.
I'm intrigued. How do they do it?
Well, when tissue is injured, your immune system sends in a cleanup crew of white blood cells called macrophages.
Oh.
Initially, these macrophages adopt a state known as the M1 phenotype.
M1. What does that mean in practical terms?
Think of M1 macrophages as the demolition team. They are highly aggressive. They release harsh chemicals that cause swelling, redness, and pain, all just to clear out dead bacteria and cellular debris.
Which, to be fair, is necessary right after the injury occurs.
Oh, it is essential. You need that initial cleanup. But the problem with severe trauma is that the macrophages get stuck in this M1 demolition mode. They stay aggressive for weeks or even months, bathing the tissue in inflammatory chemicals.
And that's the chronic inflammation.
Right, and this chronic inflammation prevents new tissue from forming, and it can even start killing off healthy surrounding cells. We call that process secondary necrosis.
So how on earth does a tiny nanobubble stop an aggressive, full-blown immune response?
It forces a chemical identity change. When the exosomes drift into the inflamed tissue, they are literally swallowed by these aggressive M1 macrophages.
Like a Trojan horse.
Exactly like a Trojan horse. Once inside, the exosomes release a specific genetic code that alters the surface receptors of the macrophage, forcing it to transition into an entirely different state, known as the M2 phenotype.
And what does an M2 macrophage do differently?
It is the exact opposite of M1. M2 macrophages are the construction managers. They secrete anti-inflammatory chemicals that actually calm the surrounding tissue.
Oh, wow. So they flip the switch from demolition to construction.
Yes, they stop the swelling, halt the secondary necrosis of the muscle fibers, and completely clear the way for all that new collagen and blood vessel growth we just discussed. The exosomes literally drop a code that rewires the local immune system from a hostile, destructive environment into a calm, regenerative one.
Here's where it gets really interesting. Because with all these incredible nanoscale mechanisms proven to work on a cellular level, how is this actually moving from a petri dish into a real patient? Because as someone who follows medical technology closely, whenever something this cutting edge emerges, the commercial market often races way ahead of the clinical reality.
That is a very, very important distinction to make. How we deliver these exosomes to the traumatized tissue is currently a field of rapid bioengineering. Because exosomes are microscopic particles rather than live tissue, the application can actually be minimally invasive.
So no big surgeries.
Right. For treating deeper introital laxity and stress urinary incontinence, clinicians are currently utilizing microinjections. They map out the damaged areas of the vaginal wall and the periurethral space, and they basically inject a concentrated liquid suspension of exosomes directly into the tissue.
But wait, if they just inject a liquid, doesn't it just wash away into the bloodstream before the cells can absorb all those microRNAs?
That is the exact problem researchers are solving right now with something called advanced hydrogels. Instead of a simple liquid injection, bioengineers are loading these exosome payloads into specialized biodegradable gels or even microscopic structures called collagen nanoyarn scaffolds.
Nanoyarn, that sounds like microscopic knitting.
It behaves very much like that, actually. Imagine a tiny dissolvable silk sponge loaded with exosomes. When injected, this hydrogel anchors itself physically to the damaged muscle and vaginal wall.
So it stays put.
Exactly. And over the course of several weeks, as the gel slowly dissolves, it provides a steady, sustained release of the exosomes. This ensures that the local fibroblasts and muscle stem cells receive a constant stream of regenerative software rather than just a single flood that disappears in an hour.
Okay, that seriously sounds like science fiction made real. But we do have to talk about the regulatory reality. If I am a listener and I see a local clinic advertising a miraculous exosome therapy to, tighten the pelvic floor, what is the actual medical consensus on this right now?
The regulatory landscape definitely requires serious attention. We can look at the framework provided by the European Medicines Agency, or the EMA, as a prime example here.
How do they classify this?
Under the EMA, treatments that utilize genes, tissues, or cells and the products derived directly from them, like exosomes, they're classified as Advanced Therapy Medicinal Products, or ATMPs.
Meaning they are heavily regulated and require rigorous clinical trials.
Precisely. Health authorities worldwide are impartially reporting a significant rise in unregulated clinics offering exosome therapies that have entirely bypassed these rigorous clinical trials. The authorities issue a very clear warning. While the preclinical data showing how exosomes rebuild collagen and muscle is incredibly promising, unregulated commercial products pose real, tangible risks.
Because you don't actually know what's in the vial. It might not be purified or it might not be the right dosage.
Exactly. Or it might completely lack the specific microRNAs needed for actual tissue repair. Furthermore, clinical guidelines emphasize that a comprehensive medical consultation is required before any treatment. Mild to moderate introital laxity and urinary symptoms can stem from multiple mechanical or neurological causes.
So exosomes are not a magic bullet for everyone.
Right. In many cases, targeted pelvic floor physical therapy is the necessary frontline intervention, or it must be combined with any potential regenerative approach. Patients are strongly advised to verify that any exosome treatment they consider is part of an authorized, regulated clinical trial.
So what does this all mean? If we step back and look at the whole picture for you, the listener, the mechanical trauma of childbirth isn't just a simple stretch. It is a profound, microscopic dismantling of the pelvic floor's architecture. We're talking snapped collagen netting, torn pubocostal muscle fibers, stretched nerves, and chronic aggressive inflammation. And for a long time, we thought we had to inject live stem cells to fix it, only to find out those cells were highly unstable, prone to immune attacks, and incredibly hard to keep alive.
But by studying how those stem cells actually communicate, science bypassed the bottleneck entirely.
Exactly. Instead of the cell, we extract the exosome, the tiny, stable, non-living flash drive. We freeze dry it, suspend it in advanced hydrogels, and deliver it directly to the trauma site. And once inside, it unleashes this highly coordinated software update. It drops genetic stop signs to halt the enzymes destroying your collagen and green lights to build fresh, elastic tissue.
It wakes up dormant reserve cells to stitch together torn muscles.
Right. It sprouts new blood vessels for oxygen, insulates damaged nerve fibers, and literally reprograms aggressive immune cells into a restorative cleanup crew.
It represents a fundamental evolution in medicine. We are moving away from blunt mechanical fixes and risky live cell grafts, and we're entering an era where we simply supply the body's own cells with the exact epigenetic software they need to heal themselves.
Which leaves us with a final, really lingering thought to explore. We've seen how exosomes act as delivery vehicles for incredibly specific microRNAs like miR93 to rewind the clock on the mechanical trauma of childbirth. Now, right now, we extract these from stem cells. But if we know the exact chemical code required to thicken vaginal tissue and fortify muscle, could we eventually synthesize and mass-produce these exact microRNA cocktails in a lab?
That is the next frontier.
Imagine a future where we don't wait for the severe damage of childbirth to occur. Could the future of obstetrics involve delivering a targeted dose of synthetic cellular armor to the pelvic floor in the weeks before delivery? Literally entirely preventing the micro tears and collagen degradation we just spent this whole time learning how to fix. When you think about it, it really makes you wonder just how far this biological software can take us.